Good afternoon. My name is Christoph Geis, and together with my co-chair, Professor Beth Notar, Trinity's Institute for Interdisciplinary Studies would welcome you to today's webinar, which is co-sponsored by Trinity's Biology Department. Before I hand it off to Professor Lisa Foster, who will introduce the speaker, I would just briefly like to outline the technicalities. You can ask questions through the question and answer button at the bottom of the screen, and Professors Notar and Foster will forward those questions to the speaker. And I think Kari has planned on a few breaks in her presentation, and then those questions will be forwarded. That's all I have to say. Lisa, it's all you, and enjoy the seminar. Thank you.
Well, thank you, Christoph, and welcome everyone. It's, one of the great pleasures of being a faculty member is the opportunity to introduce a former student in a setting such as this. And Kari graduated with honors in biology in 2003. And she made the most of her time as an undergraduate here at Trinity. She wrote a grant to the American Society for Microbiology for undergraduate research that was funded her research in my lab for a year, and then also provided travel for her to attend the American Society for Microbiology meeting. And then after Trinity, she went to the Albert Einstein College of Medicine, where she earned her PhD in microbiology and immunology. There she worked in a lab about developing a new vaccine for tuberculosis. And it was pretty interesting that she chose that lab, since one of her final papers that she wrote at Trinity was a mock grant proposal in the senior seminar class that I teach, in which her thesis was developing a new vaccine for tuberculosis. And then she goes to graduate school and kind of does that. She stayed at Albert Einstein in that same lab for her postdoctoral fellowship, and is now working as a research scientist and associate director at Pfizer. And today she's going to tell us about some of the work that's been done to help develop new vaccines for COVID. And I will the title of her talk is The Vaccine Pipeline, Developing Safe and Efficacious Vaccines to Protect Against Some of Our World's Most Serious Threats. Take it away, Kari.
Good afternoon, everyone. I hope you can hear me. And I will start sharing my screen so we can start the presentation. So I wanted to take the opportunity to talk to everyone today about vaccines, a vaccine pipeline, and how we actually go about developing these vaccines so that they are safe and effective to protect the population against, like I said, some of the world's most serious threats. In particular, the most serious thing that's affecting us right now is the COVID-19, the coronavirus. So I know it's a bit of a mixed population group. So I wanted to kind of break it down a bit and go through some of the details about what a vaccine is for people who might not be so familiar with some of the basics about vaccinology and what we do. So what is a vaccine? It is actually a product that stimulates a person's immune system to produce immunity to a specific disease. And the overall intent is to protect that person from that disease. So some vaccines you're given as a child, as a baby, and that protects you for life. Some vaccines are given as a child and you're needed to get a booster, potentially every 10 years or so. You might know this through your tetanus vaccine, that you have to get a booster every time you either step on a rusty nail or something like that. So depending on the type of vaccine, it's meant to protect you for the remainder of your life. So how does it work? Now, some of you might know about the immune response and the immune system, but for those of you who don't, what happens is you're given a small amount of a harmless form of the disease. And I'll go through a little bit later in the talk what specifically we put in the vaccines, what we're looking at, different types of materials that go in there, based on the disease we're targeting. So you're given a small amount of that, a harmless form of disease that's safe, and it induces your body to make, in this case, this example is just antibodies, to fight off the disease. So when you encounter it again after you're vaccinated, your body is already aware of it and already knows what to do. And it can mount a rapid defense against that disease and protect you so you never get sick in the first place. So that's a big difference between a treatment and a vaccine. You might be hearing a lot of things about treatments that are being developed for the COVID virus right now. But the thing is, if you are looking at a treatment, that means you're sick and you have symptoms. So with the vaccine, the intention is for you to never get sick, never have those symptoms, never have that effect, and never spread to other people. So that's the big driver of why vaccines are so important, in addition to the treatments and everything else, because we can't always protect all of the population all of the time. So let's go a little deeper about how this actually works. So some of you might be hearing in the news about things that are getting discussed about what's being induced in some of these vaccines. So you might be hearing about antibodies, about neutralizing antibodies, about T cell responses. Now, what does that all mean? So ultimately, a vaccine should stimulate a sufficient amount of lymphocytes. So those are the cells within your body that fight the disease. And there are different types of cells for different types of diseases. So you might have more of an antibody-mediated response if you're looking to fight something like a bacterial infection, some viral infections, or even like an infection like a Lyme disease, where you want an antibody-mediated response. Now, there's a second type of response called a cell-mediated response, and that fights things that get inside your cells, like viruses and parasites and things like that. So the cell-mediated response is more targeted to that. So depending on what you want to stimulate is what we'll actually look to target and try to induce in that response, which is why you hear talk of antibodies and T cells. So the second immunization, you might be hearing about a boost. Let me just, there we go. You might be hearing about a boost in the news. Oops, sorry. So what that is, this antigen second exposure is meant to really hone that response, hone that immune cell response to produce memory. And that's really important in having long-lasting immunity to the disease. So this primary exposure that you give that you've gotten in your first vaccination will train that response, will produce some of these T cells, but then that second exposure is really going to hone that response to be very specific, very effective, and be long-lasting to produce this memory. So what's different is antibody versus the cell-mediated, depending on the disease. So now when we're talking about something like COVID or coronavirus, or even something like the flu, what we're looking for is both neutralizing antibodies, which are specifically made to prevent the virus from ever entering your cells. So that means that you're exposed to it. The virus has to get into your cells to grow and to produce and to replicate and to be able to give you symptoms and spread. So these neutralizing antibodies are meant to prevent that from ever happening. Now, we can't prevent 100% of everything all of the time. So you also want to have this secondary kind of backup response of your cell-mediated response, your T cell response, which will fight and kill those cells that get infected by the virus. So you have a double whammy of preventing infection of your cells from that virus that you encounter. And then if that virus does slip through, those cells are then eliminated from your body and that infection can never spread. So that's what's really important about looking at these neutralizing antibodies, which prevent entry of the virus into your cells, and this T cell response that we talk about. Now, not every type of vaccine induces both of these, which is very important in terms of selecting the type of material that we give you for vaccination. So what are the types of vaccines? Again, you might be hearing a lot about many different varieties of vaccines that are being developed right now. essentially we are trying to throw the book at this disease right now to just try to clear it from our lives, get us back to normal. So what are the types of vaccines that are available right now? There's live attenuated, which means that it is virus, but it does not cause symptoms. There is inactivated, which is a killed virus. So in live attenuated, that virus could replicate, you could shed it, but it won't give you symptoms. In inactivated, that there is that virus, it will not replicate, it will not give you symptoms. That's why we call it a killed virus. There's a subunit, which is just portions of that virus. In this case, it can be a lot of protein that we use for immunization. And that's just a subset of the virus to really target that response and hone that response. There's, in the case of certain diseases, such as tetanus or botulism, those organisms produce toxins, right? So we can take that toxin and inactivate it. And that toxin then serves as an antigen to provide an immune response and not get you infected with that particular disease. And then there's what a lot of companies are working on now, which we consider the next generation of vaccines, nucleic acids. So all of our cells are made up of building blocks called nucleic acids. All of our cells contain DNA, which is the building block and what all of our genetic code is written from. That DNA is translated into RNA, which then makes the proteins. So that's the functional part of the nucleic acid system versus the DNA, which is kind of the library book, which contains all of the information, versus the card catalog, which is kind of the RNA, which retrieves that information and produces it and provides it to the next person. So you can see I have a bunch of examples of different vaccinations that use all of these different types of strategies. So the whole virus, portions of the virus, and then this nucleic acid part of the virus. And all of these that I have here are actually approved vaccines. So all of the, most of the children that are born in the U.S. Get these series of vaccines during their first few years of life to protect them from all of these diseases, which is why we don't have a lot of this disease prevalence in the population right now, because we have these vaccines. What we're focusing on, or a lot of companies are focusing on, is this next generation of vaccines because a few reasons. So if I go to the next slide. In this case, I'll show you pros and cons of each. This next generation of vaccines are very easy to deliver. They're easy to manufacture. They're fast. We can make them very quickly. And they induce a very good immune response, both that cellular response, which will kill that virus that's inside the cell, and that neutralizing response, which will prevent that virus from ever infecting the cell, which is why we really like these types of vaccines right now. And we're really focused on getting those to the public. There are some drawbacks to these in terms of transportation, storage conditions, things like that. Those are being worked through because it is a new technology. So as our knowledge grows and our ability to work with this and manufacture this and produce this in large quantities, all of that induces us to rapidly evolve to get the material more stable, more easily transportable, all of that. Transportation and storage stability of all of these, what we call older vaccines, they've had 50, 60 years to really evolve. So we're trying to evolve this next generation vaccines in the span of a single year. So that's one of the issues we have right now. But for all the other vaccines, there's pluses and minuses. Like I said, you can have high immunity with certain vaccines, such as this live virus. But it is also expensive to make because it is live virus and you have to grow it and incubate it. it can be difficult to produce, and it might have safety risks. So we have a very diverse population in the world, and not everyone is able to get this live virus vaccination, which is why some of these vaccinations are more targeted to subunits, such as portions of the vaccine, portions of the disease-causing organism, where they would be much more safe. They're easy to produce, low toxicity, so they won't impact the person who gets the virus, but they could have a little bit less immunogenicity. So that's partly what we look at in terms of developing those products, to make sure they are the most immunogenic, the most effective. Okay, so I had mentioned that at least for the newest next generation of vaccines that we're talking about, some of the storage and transportation conditions are what we're working through right now. But there are many options. So for all of the vaccines that are currently available in people right now, you can have something such as a multi-dose vial, where we have multiple doses that can be pulled from a single vial. This is very effective in being used in locations where you might not have as much easy access to hospitals and to facilities and to doctors' offices. So typically those doses, those multi-dose vials, are brought into communities and vaccinate the entire community from that vial. So that helps those types of communities. Single dose vials, obviously a single dose in a single vial, single shot in a single vial, which is what we're working with currently right now. The vaccine image can be very different. So it could be a liquid like you see here, just stored at normal refrigerated temperature. It could be stored frozen, which is currently what some of the coronavirus vaccines now are, stored frozen. Or it could be lyophilized, freeze-dried. There are a lot of vaccines that are freeze-dried into powders, where if they're not as stable in a liquid form in the refrigerator, once you freeze dry it into a powder, you can stick that on a shelf and it is stable for years. So in this case, once you pull this vial, you just add your diluent to it and you have your vaccination, and it is as stable as if you had it in the freezer. And then there are a variety of different deliveries systems that you can have a vaccine. So you can have a syringe delivery system. So that's your typical intramuscular shot, where they give you a shot in the arm or in the leg. You have oral vaccines, where it's a liquid that you swallow. That one of the examples is the oral polio vaccine that babies get. You can have a spray. For example, Flu Mist is a spray. So that's just a spray up your nose to vaccinate you with that. Or there's a new technology that's coming about. It's called a microneedle patch. So essentially what this is, your doctor gives you a little patch that you put on your skin. You leave it on there for an hour, you take it off, and that's your vaccination. So those are actually very, this technology is very up and coming and important, again, for those communities that don't have so much access to syringes and vials and needles, storage facilities where they can store these vials in a refrigerated containment. If they get a big delivery of just patches, they're stable at whatever temperature, room temperature, and you can put them on, and it's very easily delivered. So I think before I go into the next section, just kind of walking through how we actually go through our timeline and how long it takes to develop a vaccine. I think I want to just pause, see if there's any questions about what I've gone through so far.
Actually, Kari, there was one question that's related to this. Someone asks, is there a cost difference among the different types of vaccines?
So a cost in production, or a cost in terms of person, depending on your healthcare coverage or things like that. So in terms of production, yes, there is a cost. So lyophilizing, freeze drying a vaccine is more expensive than having it as a liquid or as a frozen vial. Yes, but that can be made up in terms of the benefit that you get in that that material is stable for longer. So that cost benefit is countered by that shelf life stability. So meaning that if you have to produce a certain number of doses and manufacture that at regular intervals versus produce one big batch of doses in a single manufacturing instance and then have that sit on the shelf, that cost gets kind of offset. So it depends on the vaccine, but there's also always pluses and minuses to each. Microneedle patches, because they're more newer technology and not as widely manufactured, they are more expensive. But as that technology grows and gets more widespread, that cost comes down. Because again, we find ways to manufacture them quicker, faster, in bulk, and then that cost comes down. So yes, there are differences, but in the end, you know, things kind of even out.
One other one that is relevant at this point is what is used as the medium for a powder vaccine?
So when you lyophilize a vaccine, meaning freeze drying, you have your liquid vial, and that goes into specific equipment. And essentially all of the water is sucked out of that vial. So if that vaccine is in a sugar water solution, which is typical of a lot of our vaccines, sugar water solutions, all of the water gets sucked out. So it's essentially just a freeze-dried powder that contains a vaccine, some sugar, maybe some fat to kind of make it a nice little cake shape. But then the water gets added back when we call it reconstituting. So when you dissolve the powder, that water gets added back.
Okay.
Okay. Okay, so I will move on. All right. So how long does it take us to make vaccines? The typical life cycle is 10 to 15 years, which is probably why you don't hear very often about new vaccines that are coming out on the market. It's a very slow, methodical process that we go through. There's typically about 4 to 5 years that we spend in discovery and early development of the material before that material even gets into people. In that interim, once we feel like we've met a certain bar in terms of effectiveness of the product within our either animal models or other measures that we take of the vaccine, then we decide we want to test it in people and humans. And that's called our phase 1 study. So our first study is safety and tolerability to make sure that we have no adverse events in people. This is after our first animal safety study. So we go through all of that first just to make sure that there's nothing amiss with the virus before it even gets to people. So that first human test is a small group of for safety. They're all healthy individuals. And then once we pass that, we typically move on to a phase 2 study where we're looking at dose levels. We're looking at we call it a proof of mechanism, meaning that we know what type of response is going to be induced in people. And then we want to also look for some early signs of efficacy, that it's effective. We can't state in this phase 2 study whether it's effective or not because it's not enough people in the study. But we want to essentially prove that our proof of concept works. This whole process usually takes about 3 to 5 years. Once we have our proof of concept that, okay, yes, what we think is going on in people once we vaccinate is going on, we think it's going to be effective and we know it's safe. then we move on to this phase 3 testing, which is those thousands and thousands of people that we test in. So in that case, that's where we're really looking for that efficacy level. So we're looking for our statisticians to look at disease prevalence, to look at the population it affects, so that we can model and say, okay, we have to vaccinate 40,000 people or 50,000 people because we know that there's going to be a certain number of people that get the disease. And we want to see that in those certain amount of cases, we have a very high confidence level that if we see, let's say 50% of those cases had the placebo and 50% have the vaccine, we're pretty sure that that's going to be representative of the entire population. So we have multiple statisticians working on this, modeling this, and refining it as we go. Sometimes we have to increase study sizes because we're not seeing the type of cases or the number of cases we need to have assurances that we're going to hit that final mark where we're seeing 50%, 60%, 70%. Sometimes we start these studies and we don't see any efficacy at all, and then those studies get stopped because we don't see any effectiveness there. So this process can take a while, depending on how things are going, how prevalent the disease is, and how protective it looks. So again, 3 to 5 years. Once we show at the end, we hit our endpoints and we say, okay, it looks effective, then we apply for licensure and then commercial use. And then we have post-marketing commitments, which are, we call it phase 4. So that means our clinical trials never really end. They just keep on going because we do have all of these post-marketing commitments. And through all of this, we connect very closely with the regulators through each of these phases and through each of these steps. So 10 to 15 years seems like a lot of time. What do we do? Why does it take so long? What do we do specifically? What are we looking for? So I guess right off the bat, how do we pick our targets? How do we pick what we're going to work on and what we're going to look for in terms of vaccination? So a bunch of things go into this. We like to look to see what the disease prevalence is. We look throughout the world and say, okay, we're seeing a lot of infection in Europe or the U.S. or in India or in Africa. And we want to make sure that the disease burden is enough to make it worthwhile to expend the resources and to make it worthwhile to protect as many people as we can. We like to look at the populations that are impacted because each type of population presents its own challenges. So if pregnant women are more affected by the disease, or if elderly are more affected by the disease, that really factors into our decisions about how we go about this, because we want to make sure it's going to be safe and it's going to be tolerated and it's going to be effective to protect people. We want to make sure that we can actually work with the organism. Some infectious diseases are not culturable. We can't grow it in a lab. So if we can't grow it in a lab and we can't study it, then we can't make a vaccine to protect against it, unfortunately. So we want to make sure we can work with it in the lab. We look at the current status of the research. So we do multiple passes through different scientific journals, academic institutions, see where the research is, see what's going on, see what people have found already. And if we see enough of progress in that, then that piques our interest about targeting that particular disease. And then we also like to see if there's some previous precedent. So we might have some vaccinations that are on the market that might not be as effective, or it might not be effective anymore because that disease has kind of waned and something else has taken its place. So we also want to see that. We want to see if there's previous precedent that we have a vaccine for example, the flu, but it changes every year. So then we can build on the vaccine from last year, change it, adapt it, and then put it out for the next season. So we look to see that. Based on all of this information and data, then we select our disease targets. So what do we do during this discovery and early development stage? We do a lot. And part of the reason why I'm going through all of this and all of this work we go through is I really want to emphasize that there's a lot of discussion about the speed at which we're working through things and the speed at which we're moving things and getting things to the public. There's a lot of steps that go into this that usually take years. And I want to just make sure that everyone has an appreciation of everything that goes into it to show how meticulous and careful and how much work and how much study we put into it to make sure that it's going to be safe and effective. So we look at the process. We want to make sure we can make it consistently in accordance with all of our regulatory agencies and regulatory guidelines, and not just within the US. There are different requirements within different countries throughout the world. So we want to make sure that whatever we're adapting for the US can be applicable to other countries, or make sure we're covering things with other countries. We want to, can we make it with previously known components? That's the easiest way. If we have material that's already been in people, we know it's safe, then that's the easiest way. Sugar water solutions, things like that. We want to make sure that we know how people are going to react to things and that it's going to be innocuous and safe. That's our biggest goal. So we don't like to put very new things in if we don't know how people are going to respond to it. And we want to make sure we can scale it up. You know, it's easy to make something in a lab in a small scale, but it's very different to start making billions of doses of something to provide enough material to vaccinate a world population. In terms of analytics, we want to make sure we can fully characterize material. We know everything that goes into our vaccines, each and every little thing that goes in, and we monitor it and we carefully track it over time. So we know how that material behaves, what it looks like, and how it looks like in terms of different environments and conditions and temperatures. We are very thorough about that. We want to understand the levels of all the residuals in the product. Again, if we have some sugar in there, if we have some fat, how much is in there? Can we monitor that? Do we see if that changes over time? We want to make sure nothing is changing over time. That's our big thing. Can we qualify and validate all of our methods? So anything that we test, we have to provide all of that information to the regulatory agencies to make sure that our tests are reproducible, not just in our labs, but in any lab that will take the material and test it. So that's very important. I'll close this. Stability. We want to make sure that our material is stable. Again, we don't want it changing because if our material changes, then that could mean that is less effective. So we want to make sure it is absolutely stable and we know what is critical for that material to be effective because we don't want that changing. And we want to know how we can improve that stability. If we don't have enough data to say that we know it's stable at refrigerated temperatures, can we freeze it? Since we know if we freeze something, then it's going to be stable. Immunogenicity, we want to make sure we're testing in the right models, and we want to show that it is relevant. It gives you relevant protection and relevant immunogenicity. That's the discussion that I had earlier about those neutralizing antibodies. You can produce thousands and thousands of antibodies, but if none of them are neutralizing, they're not going to protect, at least for certain diseases. So we want to make sure we're showing relevant protection and that it is going to be effective in protecting the population that we vaccinate. And then safety. Safety is a big thing that we look at. We do multiple toxicology studies. We want to make sure that there's going to be no adverse effects in any part of the body that we see. Again, we have to select appropriate animal models for this so that we're sure we're covering everything. And again, for safety, we like to use material that's previously been in population and people for years and years and years, because again, we try not to do anything new because we have that precedent and that safety precedent. And we want to make sure we manufacture and release material according to all of our regulatory guidelines. So all of our manufacturing is fully documented, fully written down, fully accessible. We have to provide everything to the regulatory agency, and they look through everything to make sure that we've crossed all of our I's. Crossed all our T's, dot all of our I's. Nothing gets through. Okay. Okay. So just a little bit more, I've tried to put up some pretty pictures to talk through this. So process-wise, again, we have to be able to make it consistently. You've probably seen a lot of these pictures here of those vials being filled. Even the vials, we have to make sure that we can make those vials, and we don't have a shortage of vials, glass vials, if that's what our material is going to be presented in. So there's a lot of work that goes in, and each portion of this process is covered by multiple people, groups of people that work on this specifically, looking at each of these steps in each of these areas. In terms of testing, again, we want to make sure all of our components are thoroughly analyzed and monitored using components that have already been in people. We map everything, we characterize everything fully to make sure we know as much as we can, if not everything, about the material that we're putting in. We want to make sure it's stable. We don't want our material changing as it sits on the shelf, because when it changes, then that means that it could be less effective. So we don't want that happening. We want to make sure we know it's stable, we know how to make it stable. And if it's not stable in one form, at a certain temperature, refrigerated temperature versus just sitting on the shelf, an ambient temperature, then maybe we have to freeze it, or maybe we have to freeze dry it to make sure it's stable. Go through. Animal models. You might think that testing in animals is either a fairly easy thing. We take some mice, we inject it, and we see what's going on. So mice are, we want to make sure that the animal model is representative to what we're going to see in people. So it could be different. We might have to test something in a rat, a cotton rat, or some other type of model like a monkey, just to make sure that we know everything that's going to happen within that person. Make sure we know how much to give the animal and give people, when to look and when to monitor it in terms of days, when to give that boost dose, which is very critical to get that long-term memory, that long-term response. So figuring out when to give this second dose, or if we need it. Some vaccines, you don't need a second dose, but a lot of them do. So figuring that timing out, and then making sure that what we're monitoring and we're measuring in terms of immune response is relevant. We like to take, and you might have seen this and talked about this, human convalescent sera, meaning that we take blood from people that have already been infected with the disease, and we see what their immune response looks like, and we compare it to what we're getting within our animals. And we want to make sure we're above that. Again, because the better immune response you induce, the more likely it'll be a long-lasting protection. And then safety. We make sure that we do all of our toxicology studies, animal studies, prior to anything getting into people, because we want to make sure that we know how it's going to react in every single organ of that animal. That we don't miss a thing. We want to make sure that we know how that dose behaves, how much we're giving the animal. And then we want to estimate an initial safe dose and what we want to go into people with, and identify, if at all, if there's going to be any potential adverse effects. If we see anything in animals, then it does not move into people. This is our first benchmark that we also go through. So I think, let me just pause there, see if there are any questions before I move on to the next little section.
There's been quite a few questions about pretty much about the animals that were were there animals used in the COVID testing? Which animals were used there? And then about recruiting human subjects to be part of these vaccine trials. How do you do that, and what kind of information are they given?
Yeah, so at least for most of the coronavirus vaccines that are being tested right now, the animal models that have been used are mice and monkeys that have been the benchmark for this particular disease. So we've seen a very good correlation with what we're seeing in mice, translate to what we see in monkeys, and then translate to what we see in humans. So we have a very good idea that if something shows a good response in a mouse, it'll look good in a human. So those are the animals that we've been using. And then in terms of recruiting for our clinical trials, so we have teams that are involved in that. Most of these are run out of universities and teaching hospitals and things like that. So those individuals are, given all of the information about what we're doing, what we're looking for, what we're going to do in terms of visits, doctor's visits, blood, things like that, getting the vaccine, what we've seen so far. So everyone is fully informed going into all of these studies. And we only take volunteers for all of our studies. Now we don't, none of these, we don't challenge people like we might animals. We might, if we want to look for protection, for example, in some of the coronavirus vaccines, such as the Oxford vaccine that's out there right now, they challenged their monkeys, they vaccinated them and then challenged them to look to see if they got protection. We can't do that in people. We just have to vaccinate them and sit and wait and see if they get the disease just from their normal everyday life exposure. That's typically how we run those.
Thank you, Kari. That covered a great number of questions all in one answer.
Okay.
There is another question, though it's related to the stuff you were just talking about. And it's, how are regulatory and business officials able to be so confident that a vaccine going from unknown to production in 9 months are just as safe as a vaccine that we've had around for decades, like the flu vaccine?
Yeah, so that's part of why I want to go through all of these steps about how we move a vaccine from that discovery phase to phase 1, 2, 3, and then to licensure. So from everything that I've talked about so far up to getting into people, it's very involved, very intricate, right? And usually it takes a few years. So I'll talk about it a little bit later about how we're actually doing it so quickly. It's a matter of people and resources. So we don't skip any steps. We do everything that we do for a normal vaccine and a normal product production and launch. We just put more people and resources on it. So typically where we're supporting, 5, 6, 7 different programs, we're supporting the single program. So it's thousands of people working on this day in, 24 hours a day, 7 days a week, with as many resources as we need to get it done and to make sure we don't miss anything and we don't skip anything. So like I've discussed about the animal studies, like I've discussed about using materials that have already been in people that we know are safe. Again, we try to base all of our work off of things that we already know. So it's never a completely new or novel thing that we're working on. And there's also actually some precedent for RNA vaccines. Nothing's been commercialized yet in terms of being licensed, but these vaccines have been in people before. Mostly to treat oncology or as therapeutics for certain diseases. So these have been in people. So we already have some of that experience. So we try to build on all the experience we have, all the knowledge we have from multiple programs and everything that we've done to make sure we hit everything and assure that it's going to be safe when it goes into people.
One last question I'll ask at this point from all of the questions that comes in is, what part of the process are you most involved in?
So for the coronavirus vaccine, it's been a sort of unique experience because, again, we're trying to move so quickly. Typically, I'm involved in this discovery and early development phase, and then up to just about phase 1 of material, getting it into people. But because of how we're doing this program and how we're building this kind of infrastructure around it, I've been involved in all of these aspects. So I'm on the manufacturing side of things. I don't do the clinical study side of things, the serology testing or that sort of thing, but I'm involved in all of the manufacturing, all of that work, looking at the targets, making the material, making sure we can store it, ship it, transport it, making sure it's effective, even in our animal model. So that's where I'm involved.
Oh, and you can imagine we have many, many questions. So I'm trying to, so here's one that I think is related to what you've recently been discussing. In a vaccine trial, are the side effects known before efficacy is proven?
Yes. So, well, yes and no. So if it's a completely novel material, we might not know. We have some precedent for this particular virus because of other viruses we've encountered. So other SARS viruses. So we already know what to look for. We have some precedent based on what we have in the vaccine, in the material. We know that sometimes people get, a sore arm, redness, swelling. That's pretty consistent for any type of vaccine you get. Occasionally people will get fever and chills. Again, that's pretty consistent with any type of vaccine that you get. The levels, we might not know, and we kind of dial that in that first round where we're looking at dosage. So in our first phase, you might have seen that we were testing a wide range of doses. Moderna settled on that 100 nanogram dose. We actually dialed ours back to the 30 because we saw what sort of side effects in terms of the fever, the chills, and things that make people uncomfortable but are transient, and wanted to dial that back to minimize that and reduce that as much as possible. So we have an idea going in about what it's going to look like. We have to dial that in a bit when we look at the dose and we look at the effectiveness of the vaccine, kind of weigh both the effectiveness and the unwanted side effects and pick that, sweet spot that we find. So yes.
Okay, why don't you continue moving on, and then we will get to as many of these questions as we can.
Okay. And I hope I address some of them too as I roll through, because some of this I'm going to go through. So once we get into people, what are we doing? What are we looking for? So we have, as people probably have heard, phase 1, phase 2, phase 1/2, phase 2/3, phase 3. All of these have different goals in mind, different sizes, different targets. So in a phase 1 and a phase 2 study, what we're looking for is safety and an early read on efficacy and mechanism so that we can get to proof of concept, meaning that we know it's safe. We have an idea, we think that it might be effective, and that we know how people are going to respond to it in terms of what type of antibodies, whether we're getting those neutralizing antibodies or we're getting those cell responses, those T cell responses. And we can prove that, yes, what we see in our animals, we're seeing in people, and we feel confident to move to the next round. So phase 1 is going to be a small population of healthy individuals. This might not be the target population. So like I said, if there's a vaccine that you're looking for to immunize pregnant women with, because there's a particular disease that affects them, then we do not go into those individuals in our phase 1. These are just going to be healthy adults between the ages of 18 and 45, depending on the trial. We want to make sure that the material is safe in those populations before we try it in any other potentially compromised or any other population that might have other comorbidities or other issues. This is several months, the duration, because we do that first vaccination. We have specific safety readouts that are dictated by the regulatory agency, whether it's a 2-month readout, a 1-month readout, or a 6-month readout. So that is set by the regulatory agencies about what we have to meet in terms of seeing safety before they approve it to move on to the next phase. And then we look at all of the potential side effects that we see. So anything gets reported. And that might be why people have been seeing some pauses on vaccine trials. If there is anything that comes up during this study, during this phase, whether it be a headache, a migraine, you stub your toe, anything gets reported. And the study could be held. It could be completely unrelated. But because of our awareness and our concern for safety, we want to make sure we monitor that, and we pause and make sure it's not related to the material we're giving to people. So then once we see that it's safe, we can move into phase 2. Sometimes these are combined. If we have some precedent and the regulatory agencies feel like we can combine the two in a slightly larger population. So that's 100 to 300 people. This study could be up to 2 years, depending on, again, the need of the vaccine and previous precedent and what the regulatory agencies feel like would be sufficient to prove that our material is safe, and that we show some signs of efficacy, whether it be antibody levels or T cell responses. So once we can hit those bars and prove that we can produce something that's safe and we think would be effective, we've hit proof of concept. That's what it's called, proof of concept. So then we move into phase 3. And again, what we might do is either combine phase 1/2 together or combine phase 2/3 together. Again, depending on the need, depending on the precedent and what the regulatory agencies are recommending. So phase 3 is going to be thousands of people. Now, the size depends on the pervasiveness of the disease and the statistical modeling to understand our primary endpoints. Now, what does that mean? So when we propose a clinical study to the agency, we propose to them specific primary endpoints that we're looking for. So in the case of a lot of the coronavirus vaccines, what we're looking for is people that show symptoms and then test positive, PCR positive for the virus 7 or 14 days after their boost shot, so their second shot. So if we have a case that comes up after 7 days of that second shot, it's considered a positive case and it gets recorded in the study. If a case comes up in between that first and second shot, we provide support and supportive care, but it doesn't get scored in the study because it's before that boost shot where we're really seeing if we're getting memory and immunity to the virus. So depending on the pervasiveness of the disease, that modeling could mean that we have to immunize 40,000, or could mean that we have to immunize 100,000 people to hit that. So we need to hit a certain amount of cases in our study. And that's all done based on statistical modeling. So in our case, in coronavirus, because it is so pervasive, the primary endpoints are actually very small. So we have to hit 164 cases. Any of the vaccines right now have to hit 164 positive cases. And then that says that you've hit your primary endpoint of the study. Once we hit that endpoint, the results are unblinded by the agency and the committee that is reviewing and monitoring the study. So that is not the pharmaceutical companies making the vaccines. It's an independent group that's managed by the regulatory agencies of where you're conducting the study, whether it's in the U.S. or Europe or Japan or wherever. So that group looks at the data and provides that data to the company. Again, we might not see, the actual data until we've finished our endpoint. So for example, the news that came out about the effectiveness, we still haven't seen the data. All we've been told from that independent agency that's monitoring the study is that it looks greater than 90% effective. And that's based on that committee of scientists and regulators and federal agents that are on that committee. So we have no chance of looking at the data until we've finished the study. We want to try to target a very diverse population because people react differently to vaccines. Again, in this case, we try to expand it as much as we can from, it might be 16 years old to 85 years old. That gives us a wide range in age. We want to hit an ethnically diverse population. In certain cases, we might exclude participants if we have initial concerns about safety. Again, that might be the pregnant women that we would exclude from the studies, just because we don't want to get too far ahead and make sure that it's safe in a normal population. Or we might not expand the study to people that have cancer or things like that might be immunocompromised. This could take 1 to 4 years. We look at the safety, efficacy, and dosing. And by the end of the study, if we've hit that primary endpoint, the agency is satisfied with that data, then that is one part of getting our filing accepted and approved. So just because we get good results from a clinical trial doesn't necessarily mean we can get approved, because there are multiple aspects of getting that approval from the FDA or from whatever agency we're applying for. So how do we work with the agency? Now, it's very involved and lasts throughout the entire process of the vaccine, the life cycle of the vaccine. So we start with that initial, we call it a pre-IND or an IMPD. So we submit an initial filing to the regulatory agency saying, hey, we have this material, we have this product, we think it's going to be effective, we have all of this research, all of this data, we can identify the product, fully identify all of the components, understand that antigen, run through complete disclosure of all of our manufacturing processes, lay out everything we've seen in preclinical studies, and it's usually a several thousand page document that we lay everything out, all of our methods, everything. The agency usually goes through this with a very fine-tooth comb. We get multiple queries back, we have to respond, and then we have our pre-IND meeting. In that case, if they see that it has potential and they're satisfied with what they've seen, then we can submit that IND application. Now, once we submit that IND application, that means we can get into phase 1 and get into people. Every time we make material, we have to submit an amendment to them saying that we've produced this material, this is going to go into the study, here's all the details about it, everything we've seen, stability, everything. That gets sent. We can't put it into people until they go through it, they approve it. So each step of the way, we submit more and more documentation to them. Now, all of our clinical studies are monitored, again, with an independent group. Everything is double blinded, so we don't see anything that comes through. All of the data goes to that independent committee, and they make all of the decisions about what they're seeing. And then we still continue our non-clinical development studies. So anything extra that the agency asks for us, whether it be monkey challenge, whether it be additional safety studies in animals, if they want to look at additional characterization, we have to do that and submit more documentation to them. So once we run through all of their clinical studies, then we can start to move toward that BLA filing. So business license authorization. So in terms of the current material and everyone making these coronavirus vaccines right now, because of how things are going, this is a rolling submission. So typically for other programs, classic programs, we put the document together. Again, it's thousands and thousands of pages. Each section is probably 1,000 pages that we've written, and we've fully characterized and laid out everything that's gone into the study, whether it be materials, manufacturing, what we've seen in people, what we've seen in animals, all of that goes in. Because of the speed we're moving, what we do is a rolling submission right now. What everyone's doing, a rolling submission. So as we get things, we submit it. So if we're already manufacturing, for example, emergency use supplies, we're already submitting that material to get reviewed by the agencies. So they have that and they're looking at that in real time. So then once they're satisfied, they see that we have good efficacy, we have good safety, they're satisfied with our manufacturing practices, our stability of our material, our animal and everything look good, then we can get approval. And again, we have to make sure that we can make it consistently, that it's not just a one-off. We make it one time, it looks great in people, and then we can't ever make it again. So we have to make sure that we can show them we can make it consistently of the same quality, the same material. And then post-approval, that's typically where we're continuing sending them, every lot that we release, we send them. We have documentation. They have annual or biannual facility inspections that they go through. They look at our facilities. They inspect all of our documents our documentation for how we make, each little step of the way. So it's very involved, requires a lot of writing, a lot of authoring, a lot of review, and a lot of waiting to see if they have questions, and we can't move forward until they're satisfied, fully satisfied. So, okay, so I'm going to stop right here. Is there any other questions before I move on to the next little section?
Yep, there's a few more things on safety.
Okay.
So I guess there's some confusion around how you were explaining or how the system works with the other agency reviewing the data. So you said that Pfizer hasn't yet seen the data proving that it's 90% effective. So how are these decisions made? If Pfizer hasn't seen the data, how do we know that this vaccine really is effective? Who are these people that are telling us it's effective?
So it is a committee that is put together from regulatory agencies. So it is representation from the CDC, from the overall FDA organization, independent scientists. So they also tap in, like any other peer review. They tap independent scientists from different either universities or specialties that sit on the committee. It could be doctors. I don't specifically know who is on the committee. I don't have that information, but it could be, for example, government officials that sit on it as well. They look at it. It's going to be statisticians. It's going to be clinical doctors. So a whole team of people that sit on it that look at the data and look at the data as rolling in. And they've done it, you know, essentially they've done it for all of the vaccines that have ever come through. So it's that same group of people that look at it. And that's part of the way that we ensure that the pharmaceutical companies have no influence in how the data is interpreted, because it is an independent committee. So, you know, we don't want people thinking that we're getting the data and, you know, we're trying to make it look better than it actually is. You know, this is how the process typically goes. They typically tell us what they're seeing, but because we haven't reached that final endpoint, so we've hit, you know, I think our latest was 94 cases or so. Because we haven't hit that final endpoint, we can't see the data. Ethically, we can't see the data because, again, we don't want to have the appearance of undue influence in that data. So once we hit that 164, then we get to see everything. We get to kind of have a, you know, a post-action review, I guess, to see it and look at it and then understand it ourselves. But it's a whole team of scientists and outside contributors that sit and evaluate the data.
Give you one more at this point, and that's about the immunocompromised folks that maybe aren't included in the clinical trials. How do we know, and how can one assess whether or not the vaccine will be safe for people who have immunocompromising conditions, or maybe have had a transplant or something like that?
Yeah, so that's a great question. So we typically, in our clinical trials, as the trial goes along, we typically start proposing, once we have enough safety data and we see that, at least in healthy individuals and in the age range that we're looking at, it's safe, then we start proposing additional subsets, additional cohorts of groups that we want to add to it. So for example, in the coronavirus trials, you know, some of the ages targeting have been 18 to 65 or 18 or 16 to 55, you know, depending on the group that's proposing the study. So once we have enough safety data on that, then it might be expanded to the 85 range, and it might be expanded down to 12. Because again, we want to make sure that we have safety within the healthy population, and then we start expanding those cohorts. I know people are already applying to expand the studies into HIV populations, HIV positive populations. So again, it's sort of a building blocks that we want to make sure we see safety first in those healthy populations. Then we start building that kind of you know, building that bridge to that next population where we're seeing, okay, we have safety there, then let's try it in this population. Okay, we have safety there, then the next population. So that's kind of how we progress with any vaccine.
Kari, there are lots of great economics questions here, but I could wait until you're done.
Okay, yeah, I'm actually almost at the end. So what I wanted to actually just kind of talk through, and this question is coming a lot. How do we accelerate this? Because again, it usually takes 10 to 15 years to put a vaccine to market. How do we do it in 1 year? You know, essentially the answer is people and resources. You know, if you're provided with thousands of people to be able to work on, you know, a particular product, then we can get more done than, you know, 2 people working on that material. So the more people we get, the more resources we use. Again, typically, if we're working on multiple programs, we have to spread those resources out between all those programs. Because we're really focused on a single program, we can hone and focus all of those resources to be able to make sure we have all of the materials, we have all of the transportation, even moving materials from site to site, making sure we can do that in a very rapid manner. So instead of waiting for the mail to come, you know, a week later, we can get material overnight. So things like that, people and resources. We use our years of successful experience to precisely target the work that's required. So we've manufactured and licensed many vaccines in our in the lifetime of our company. So, we use all of that experience. So we know specifically what has to get done to make sure we hit all of those bars, we hit all of those levels. We don't do a lot of the extraneous, oh, that's interesting. That's an interesting question, but that takes time, and that takes time away from, this is the actual question we have to answer, and this is the actual work we have to do. So we're very focused. We don't skip any steps, but the steps are overlapped, and work occurs assuming success. So for example, we'll manufacture material at that large scale, assuming that it's going to work. So normally in our process, in that, just a manufacturing scale example, in our process, we'll manufacturing the small scale, and we characterize that material and we see what that looks like, and then, we say, let's do the next scale. We start at 100 vials, and then we'll do like 1,000 vials, and then we'll do, 5,000 vials. In this case, we're doing everything overlapping, assuming that it's all going to go well. And that's also just based on our years of experience manufacturing and knowing how to do things, knowing how to do it, and using that skill. And then also closely working with the regulatory agencies to ensure rapid feedback to our strategies and rolling data submissions. If we're doing something, that they might not be so happy about, that they don't like, we want to make sure that we can pivot and adjust how we're dealing with things and how we're doing things to address that right away and in real time, rather than in some other programs, our other typical traditional programs where we work on it, then, we pull everything together, we present a package to them, they have some concerns, then we have to readjust and work on it a little bit more and then send it back. So here we're working in real time, and that's partly enabled because the agencies really want something to move forward. So they're making the time for us, and we're making the time for them to really work back and forth. So that's in a nutshell how we're doing it so quickly. Amount of people and resources that are available to work on this is astronomical. And that's just because we want to, get something good, get something safe, effective, so everyone can go back to their regular lives. That's our end goal, to bring everyone back to normality if possible.
Kari, there are some questions that actually fit really perfectly now. There are people who are asking, they understand how this is happening so fast, so quickly, but how can you assess any long-term side effects or long-term effects of the vaccine, because the time frame in which it's being approved is so compressed?
Yeah, so again, that is circling back to using material we know, and using precedent that have already been in people and have been in people for several years. So like I said, even though RNA technology, what they say is new, in terms of vaccine, quote unquote new, it's actually been in people for multiple years now. So we know it's safe, we know it's effective. It's been treating people in terms of therapies, gene replacement therapies, things like that, and cancer treatments, oncology treatments, things like that. So we know it's safe in that case, and we can build on that knowledge there. In terms of the things that we put in the vaccine, again, we stick to what we know. We've had particular materials in there. We know, sugar water, specific fats to make sure everything stays stable. All of that is in our material, and that's been in vaccines for years and years and years. So we know safety on that. Some of the unknowns are how people are going to react to that specific dose. But that's what we do in that very first study. We want to try to dial that dose back, because again, it could be that a previous vaccine needs a little bit higher dose. This one, we want a little lower dose. So we work with that. So it's using all of that knowledge and experience from previous work, putting it into this, making sure that, everything we know in there is very well characterized, understood, and has either been in previous previous presentations to people for years, just so we can ensure that safety.
Thank you.
Kari, there's a bunch of questions about the business aspect of things. And one question was, someone was wondering if you could give an estimate of the total investment that Pfizer will have made from R&D to manufacturing to distribution, assuming approval.
It's going to be a very big number, a very big number. Again, that's because the company is putting as many resources as possible on this program to achieve success. You know, it's a very large number. I can't estimate off the top of my head right now, but it will be a very large number. Just, you know, using specific jet, you know, for example, we have our corporate jet. You know, our corporate jet has been taken out of normal service and now is in only service for COVID work. So it's transporting material back and forth to our sites. It's transporting, you know, samples. It's transporting things that we have to test, things like that. So all of our normal kind of operating has been shifted to focus on COVID work and prioritize COVID work. So it will be a very large number, very large investment.
Thank you. Someone had an interesting question too, that with the vaccine, everyone is hoping that our lives can go back to normal, but they're wondering if for vaccine development, this will become a new normal of the fast timeline. And so our lives might go back to normal, but someone's wondering, oh, will your lives not go back to normal? This will be the new normal.
Yeah, so unfortunately, if you show you can do something in a short amount of time, then that's going to be the expectation going forward. You know, there might not be the same crisis going forward with new vaccines, and there might not be the same resource availability, you know, to put on a project because of the crisis that we're in right now. So things might shift slightly back to where we were. More likely things will be more rapid. And that's partly because using this crisis, we're also understanding how we can streamline our work, you know, utilize some of the newer technologies such as Teams, for example, you know, working on Teams, having multiple people edit documents simultaneously, you know, that seems simple, but that cuts time out of things. So understanding how we can do simple things like that to cut our timelines down is really, you know, going to impact us how we work going forward.
Thank you. Maybe I'll just ask two related questions and then turn it back to Lisa, because I thought there's some more biology questions in there. That people were wondering about Pfizer's collaboration with other companies, or how does it work with, there's a question about a German company, and then there's questions about, like, does Pfizer ever say, oh, well, or do the companies who are working on vaccine development ever say, okay, you work on this part, we'll work on this part, and then, or, you know, kind of split up some of the labor or split up some of the work?
Yeah. Again, that's a great question. So we do have multiple collaborators that we work with for some of these projects. In particular, for coronavirus, there is the German company BioNTech that we work with for this particular program. So yes, again, part of this whole understanding how we can move quickly and rapidly, we have to evaluate and understand each company's skills and skill sets and techniques and what they can do and bring to the table. So, you know, all of our calendars, you know, for example, my calendar is booked from 6 a.m. until 11 a.m. with meetings with our German collaborators, you know, because that's right in their time window with the time difference. but just spending time understanding, what they do, how we can, like you said, break up the work, make it the most efficient that we can do. If we have expertise, for example, in, electron microscopy, but they have expertise in, separation or something like that, then we say, okay, we'll do these set of experiments, they'll do those, but then also we want to make sure that if we're running material at either site, or regardless of we're running it, if it's the same assay, we're going to get the same results. So we want to ensure robustness across all of our sites and all of our collaborators. And that's one of the big things. If you can't reproduce it, it's not science. That's one of the big things about peer-reviewed articles and things like that. You want to make sure you can be able to reproduce it. So that's one of the things we drive for. So yeah, it's a big partnership, collaboration, understanding, how we can do things to make things most efficient and effective.
Thank you.
Okay, Kari, so here's another group of questions about, someone asks about, does this vaccine prevent just the progression of disease, or does it prevent infection? And if it doesn't prevent infection and just progression, can people still transmit the virus, even if they themselves don't get sick?
Yeah, great questions. So we don't know yet, again, because we haven't seen the data. We have hints based on our monkey studies, and based on some of our animal studies, and not so much the phase 1 because they were too small, but some of our early preclinical work, that it prevents the infection as well as the the replication of the virus. So if you get exposed to that virus, it doesn't actually get a chance to take hold in the body. So you're never actually shedding it or replicating it. Those are the hints that we have in the animal work. We don't know how it's going to how it's going to behave in people yet, but we're hopeful that it's going to prevent the infection itself and the spread, not just the progression of, to symptoms and things like that. But we won't know until we see that full data set. And it would be different. So different vaccines have different responses. Some vaccines fully prevent infection, getting infected in the first place. Some vaccines prevent symptoms. So you can get infected and you can spread it, but you never get symptoms because it's at a low level and then your body clears it. So we won't know until we see the data.
Kari, was there anything more you wanted to go through before we just then did all the questions?
I might have, I think, yeah, 1 or 2 more slides. So, oops, sorry.
Good, bunch of questions about this.
Cold chain. So I'm sure that a lot of people have been talking about cold chain, cold storage, frozen storage, because certain vaccines are frozen because, again, we can't, the material isn't stable in a liquid, either refrigerated or at room temperature. So how do we handle cold chain and cold storage for our particular material? So again, we have other vaccines that we have that are frozen. So we've dealt with this before, but there is a bit of a unique sense in this, in that we need to make sure that we can distribute it with this cold chain in mind, because we have, at this point, because of the data we have limited a limited window in which we can provide it to people. That is because of the data we have. So as we generate and collect more data, it doesn't mean that it's not it's not stable, it's not effective, but we only have so much data to say that yes, it is good at these temperatures. So again, as we continue our studies, keep this going, we might be able to expand that window. But based on what we have now, we've been designing strategies, and we have teams working on this, designing strategies to be able to handle this cold chain, cold storage. So it goes from, our manufacturing facilities, the trucks that we use, different storage facilities, how to get it back and forth, distribution centers, and then working with those specific hospitals or retailers that are going to get and receive this vaccine. You might have heard that we've been producing specific containers that are capable of transporting the vaccine in cold storage. We're working with a company that has specific labels. So these labels go on the vials and they track the temperature. So we can read those labels, and if that vial has seen temperatures that are, like refrigerated temperatures or anything outside of our recommended frozen storage window, we will know right away. And our motto is, when in doubt, throw it out. We don't want to risk anything with this. So we have a very involved process of tracking, making sure we see, what temperatures everything is at, and then distribution, and working specifically and closely with those places that are getting it, especially the more rural locations, understanding how we can do that, how we can enable distribution there, whether it's delivery of dry ice or using these specific special containers that we're working on. And the other thing too is, we never stop working on the vaccine. In this case, I had mentioned previously about freeze drying. That's an option that we use for material that's not stable in a liquid environment. And freeze drying is a lot, gives you that flexibility, because you could transport it refrigerated, or you could transport it at room temperature and ambient temperature. But we just don't have that material made yet. Previous work, and again, like I said, we like to work with precedent and things we've done before so that we can get it rapidly to people and we know it's safe. That work is ongoing now to try to transition it so that we can store it at different temperatures. So there's constantly work ongoing to try to improve the process, get the second generation of things going as we're trying to get the first generation out. So.
And with that in mind, how long do you think what's a realistic timeline for when the Pfizer vaccine will be available to the public?
So it depends on the particular public that we're looking at. So I'm not sure if final decisions have been made regarding about a distribution hierarchy. I know that first responders and healthcare workers are going to be on the front lines of getting the vaccine, but then where things fall out from there, decisions are still being made based on multiple factors. So we already have material sitting in the freezers ready to go once we see that final readout, more probably closer to the end of the month. So we already have material that's ready to go. Once we see that final readout, once we get our, emergency use authorization, we can start distribution, immediately, at least to the frontline healthcare workers. So it's hard to say based on when we might get that readout. I think it's going to be sooner than later, just because of how many cases we're seeing now throughout the country. Probably healthcare workers, maybe end of this year, beginning of next year, and then, kind of the hierarchy down the line next year.
Thanks.
So just to follow up on that, because there are quite a few questions on this, like general public, what might be the timeline?
Yeah. So, I think, and part of the strategy with coronavirus vaccination is that multiple companies are working on it. We will try manufacturing billions of doses within the next year, but even that's, that might not be enough to vaccinate everyone that's needed, because again, it's a 2-shot series. So, that cuts the people we can reach in half. So it depends on if we see positive readouts from other companies too. The more people that have positive readouts, the quicker we can get it to the community, whether it be, Pfizer's vaccine or, the Oxford vaccine or Moderna. So that's going to help be able to distribute things to general population quicker.
With all these different vaccines, how does one decide which vaccine they should get? I mean, are there different vaccines more suitable for different populations and things like that?
I mean, I would always say Pfizer vaccine, but I'm biased. So, as long as the safety and efficacy looks good and FDA approves, it should be pretty equivalent. Going back and forth, at least at this stage between vaccines, it could be that, if we don't have enough of ours, there might be enough of the Moderna vaccine, let's say, if it comes out. Obviously, wherever you're going, healthcare providers, different pharmacies will stock certain vaccines, but it's always within your right to request a specific one. That there's a little tidbit, to if there are multiple companies making it, to request a specific one. So at least from what's out there right now, it doesn't, I think it's too early to tell whether ones will be better in specific populations or not. At least we know that ours is effective in the older populations where we really want to make sure that they get vaccinated, that 65 to 85 range. So that's important, at least to us, we think. So we'll have to see as the data keeps rolling out.
Thank you.
Kari, did you have any more slides, or if not, we can, or if you want to finish and then.
Yeah, 2 more slides. So really quickly, like I said, do we ever stop work on our products? Absolutely not. So once we get approved, we're in phase 4, which essentially never ends, at least for us. We are constantly working on it, constantly monitoring it, constantly coordinating with the regulatory agencies. So, we never stop working on something, even if we think, oh, it's sunsetting, it's, moving on to greener pastures, it still stays with you. And we still work on it. So we can constantly want to try to improve and identify and things and implement things that we can in terms of the vaccines that we make. And then finally, just a little bit of a little bit of a lesson on herd immunity and why we want people to get vaccinated. The consideration is, again, like I said, not everyone can be vaccinated. There's specific conditions that might prevent you from being able to be vaccinated, or you might be allergic to some component in the vaccine. Sometimes you get asked if you have an egg allergy, and then you can't get the specific flu vaccines because they're produced in eggs. So, not all of the population can get vaccinated. So what we try to do is, promote that enough people get vaccinated within the population to prevent those folks who are not immunized or who can't be immunized from getting infected. So, if we have in general, like what's going on now, we have a population of people, certain folks are infected, and then each of their contacts, potential contacts can get infected and then spread that virus to the next set of contacts. So you see very quickly that the disease spreads through the population. Now, if we're immunizing a small percentage of that population, 10, 20% of that population, maybe 30% of the population, you see some protected individuals, but then those infected individuals still can expose themselves to unimmunized contacts and the disease still spreads. So to get really effective protection within the population, to be able to go back to normal, we have to make sure that we have people who are immune to the disease at 80%, 70%, 80% of the population. That way that these specific individuals who can't get vaccinated for some reason, or who don't have access to it for some reason, are protected. So essentially we form a barrier around those people from the infected people. And you can see that, maybe one person gets infected, because of close proximity, but for the most part, these people that can't get immunized or aren't immunized are protected. So we really have to immunize a good portion of the population to be able to be effective at this. So this is my last slide. So I will take some more questions.
Yep. Actually, Kari, would you mind stopping your screen share?
Sure.
There we go. This way. So there have been lots and lots of questions. I mean,
Okay.
any sense of how long the immunity will last? Is there any way, and how can you assess that from the data that you have now?
Yeah, so, we have some previews again with the monkeys that we're immunizing, in terms of duration of that immunity. It gets a little bit complicated because we're not just looking at antibody levels. So you could have waning antibody levels, but still have a good memory response. So that when you get exposed again, that memory response comes roaring back and can protect you. But also it's slightly complicated because of that cell response, that T cell response. So we have hints that, at least from some of our animal studies, that it is, it has some duration. How long of a duration, we just don't know yet. And we won't know until we keep monitoring those people. So everyone who's vaccinated gets monitored for 2 years, so we'll constantly take blood from them. We'll see what they're doing, see how their response is going, and then get a better idea. At this point, because we can't predict it and we don't have the data, everyone who is making a coronavirus vaccine is suggesting it might be yearly. But that's just because we don't have the data. It's not like flu where it mutates so quickly and so rapidly. So we don't have that issue, but we just don't know how long exactly it'll last. We're hoping it'll last a while, but we just don't know.
Thank you.
Thank you so much. There's a question, another kind of economic question here about patents and how long the patent would be good for, and then would it go to a generic version?
Yeah. So certain, we can file patents on certain things. You can't file, you can file a patent on anything. It doesn't mean it's going to get approved. So, specific parts and portions that are novel to our vaccine, we will be filing patents on. But some of it already is in the public domain. So, essentially anyone is working on it and can be working on it. So we try to, file for some of our know-how in terms of making it, but we also want to, at least for this, make sure that enough companies can make it so that people, it's a global problem, that people can be protected. So, typically if you file a patent, depending on the type of patent you file, it could be 10 years, it could be 20 years. It just depends on what you're filing. And then it'll go generic after that. But I know at least for this particular problem and this particular virus, many countries are trying to make the vaccine free to their population. So in this case, it wouldn't so much matter if there is a generic available or not, because that's the biggest thing about, cost, in terms of wanting a generic over a brand label.
Thank you so much. I think we're about at time here. This has been a really great talk, and people have been writing in thank yous too for taking the time to do this with all of us today. There are a bunch of questions we didn't get to. I don't know, Lisa, if there's one more question you want to get to, or if we should just stop here.
Most of the other questions are about, is it possible, a lot of them could be grouped together about different doses of a vaccine. Is it possible that you could have different doses that would give some sort of protection, but maybe limit side effects if people are worried about that?
So that's part of what we did in our original phase 1. So you can't specifically, when you go in to get vaccinated, you can't specifically ask the doctor to give you only a little bit. It's set usually when we release that vaccine, but that's part of what we did in our dose study, where we dialed it back down to 30 nanograms, because again, we wanted to reduce those side effects, make them very mild, but also give you enough protection, which is different, could be different depending on the dose you're using.
Thank you.
Yeah.
Thank you so much. This was really fantastic, very informative, covering both the biology, the manufacturing, the research and development, the testing. You covered so much ground for us today. So it's really, thank you so much.
Yeah, of course. I think part of our desire is just to educate people, so that they feel more comfortable about what we do and the work we do and the care we take in doing that work, and how meticulous we are to make sure everything is right and everything is safe. Just to try to reassure everyone that we're working hard, and we want everyone to get back to normal. So.
All right.
And thanks to everyone for joining us today and for all your excellent questions.
Yeah, we're sorry we couldn't get to them all, but there were many, many questions.
Yeah, no problem. If there are a few questions that you want to lump together, you can always send it and see if I can field a few. Just write some responses. That could be an option if there are a lot that I didn't get to.
Well, one interesting question was, in your 15 plus years of doing this, how do you see this vaccine is going compared, and how do you see this changing the whole industry?
So this is, I always say, and whenever I talk to people about this, I am just amazed and floored about what people can do when they put, all of their effort and their mindset and their resources behind something. Just the amazing things that can be accomplished when you really want something to get done and get it done right. So it's great. I knew we could work as a team. We could, do things well. But just this crisis has really, shot things above and beyond anything I've experienced previously. And also we deal with the same things that everyone else does in terms of social distancing, in terms of mask wearing, in terms of the amount of people that are allowed on our site. So having to deal with managing, kids that are home from school that we might have to help also, or other parents, and managing all of that along with doing our work. It's been amazing to see what people can do and adapt to and work on. And I think it is definitely going to change how we do things going forward, for good. So.
Well, thank you for that. I think we should end it here.
Yep. Thank you, Kari.
Okay. Great. Thank you, everyone.